Welcome everyone and good afternoon. So I'm very pleased to introduce our Capstone students this semester. It's actually a brand new course. These are the Capstone students in the Marine Landscape Ecology course, which is one of the brand new capstones here in the Marine Science program. So just briefly, what the students were looking at is they were actually working with a pretty large regional monitoring program that monitors rocky intertidal systems here. And so they're working with them a little bit to look at some of the methods that they currently use and what are the pros and cons of moving to sort of more modern surveying techniques. So the students are going to talk a bit about what they did, what they found, and some recommendations that they might have for the program that we work with. And so please welcome the Capstone students.
Thank you, Professor. All right, so our main question going into this project was, how are measurements of species diversity influenced by a change in sampling scale? To help answer this question, we turn to the field of landscape ecology. Now, this landscape ecology is defined as the study between the relationship between ecological processes and spatial heterogeneous areas. So, by using methods from this course, as well as specialized technology, we can answer questions about ecological processes relating to spatial scales at spatial patterns, at changing scales. So the reason why scale is so crucial when designing a survey is that if your scale is too small, you're not going to have enough data. If it's too big, you're going to have too much data. So here I have an example kind of demonstrating this fact that by changing the scale just slightly, you can get a different outcome in your results. So on the left, you can see that we have a top view overlooking a quadrat on a muscle. So if you look inside, it looks like just a basic muscle bed. But if we take a cross-section of that and zoom in a little bit, you can see that it's actually much more complex and has more species present. Now, the issue of scaling is especially important when discussing, especially to sites like ours, the rocky intertidal. Now, the rocky intertidal is a very complex ecosystem that demonstrates these clear patterns of zonation, meaning that species found here are not necessarily going to be found over here, thus making it very important when determining what scale so that you don't lose what species. So this brings us back to our initial question concerning what would happen if we changed our sampling scales as far as our measurement of species diversity. So we hypothesized by changing the sample scale, we will see an effect on species diversity. And we also had a supplemental question going in, wondering the advantages of using the photo quadrats versus the point contact method.
The study was carried out at 3 sites in Monterey Bay. These sites are run by a U.S. West Coast long-term monitoring program called the Multi-Agency Rocky Intertidal Network. And we use these sites because we want to assist Marine and possibly incorporating additional methods besides the one that they use. In the first method, for the first method, we took photo quadrats and we created fishnets using GIS, similar to a traditional quadrat, which is seen in the middle photo. And for the second method, which is seen right here, we use Marine's point count method. And the small scale. The medium scale looks at 100 points along a transect line. There were 400 points total per transect per site, 4 transects per site. We use a TopCon total station, which is seen right here on the right image, to obtain the location of the photo quadrats relative to mean low or low water. And this allowed us to obtain control points to later analyze in GIS. These are the spatial scales that we use. In the small spatial scale, we took photos of photo quadrats. In the medium scale, we combined transect lines. So there were 4 transect lines, and in the large scale, it was the entire site. In the large scale, we used a new method of classifying photos. We took photos of the entire site to merge overlapping images into one panorama image using Adobe Photoshop. And then we use a Trimble GPS unit to obtain control points relative to mean low or low water. We also arrayed transect lines, so again, there were 4 transect lines in total.
All right, so after collecting data in the field, we uploaded the photo quadrats and panorama photos into GIS. And for identifying species for both photos, we used 2 methods. For the photo quadrat, that's seen on the left right here, we used a fishnet grid. Well, first we georeferenced all 5 quadrats per transect in GIS, making it resemble the transect it was a part of. Making a fishnet grid, for each grid we would identify what species was found in each grid square. And for the second method, we used an auto classification system for the whole panorama photo, that's seen on the right here, which took advantage, auto classification system took advantage of certain wavelengths certain species would reflect in the photo. So as seen as this picture, in this picture on the right here, certain colors in this pixelated image represent a certain species. And then for our statistical analysis, we used 2 t-tests. The first one compared the 2 medium scales, which were the marine point count system compared to the photo quadrat transects. And then the second t-test compared the large scale sampling scales, which was the auto-classified panoramas compared to all four photo transects in a site put together. We also ran a ANOVA test to compare all 3 sampling scales to see just the difference of species diversity between the 3 scales. So here's an example again of the small scale, which was the individual photo quadrat. There were 60 of them all together for 3 sites. Medium scale, 12 transects for all 3 sites, and then of course the panorama, there's 3 for 3 sites.
The results of our first t-test comparing Marine's point count method to our photo transect method suggest that there is a significant difference between the 2 methods. We observed a greater number of species using the photo transect method. This shows that there are advantages to transitioning from the traditional point count methods to these new photo quadrat methods. After finding that there is a difference between the survey methods, we were interested in seeing if there is a difference if the difference in sample scale had an effect on the measurement of diversity. The results of our ANOVA suggested that there is a difference between each sampling scale. As can be seen in the box plot, the average species count increased as we scaled up from small to medium. However, when we scaled up even further from medium to large, species count decreased. We could also see that variance decreased as we scaled up. We found that the medium sample scale was the optimal scale to use with these photo transect methods. The medium scale captured the highest amount of species as well as having a lower variability. We then wanted to see if we could scale up even further, because even though the photo transect method was an efficient way of collecting data in the field, the photo analysis process was tedious and time consuming. We found, however, that we are not quite ready for that yet. As you can see, the manual classification method of the photo transect method captured a far greater average of species than the auto-classified panoramas. This could be due to the software not being able to differentiate between different species of green algae or red algae, since they might reflect the same wavelengths, or the panoramas not being able to capture smaller species such as limpets or snails.
So we are testing to see if sampling scale influenced the measurements of species diversity, and we can clearly see that it did. For our particular study, we saw that the medium scale using the photo quadrats was the most optimal. This may be because compared to the marine point counts, we are able to use photo quadrats which covered a larger area, and we are able to collect more data in a shorter amount of time. And because we collected more data, we are able to be more confident in our results when looking at species diversity. So how this can be applied in marine monitoring, if organizations start to incorporate our methods of using photo quadrats, they may be able to become more efficient and collect more data over time. This could be used if an organization wanted to look at variation in species within a particular site. They would be able to survey the entire site in a shorter amount of time and would become easier to monitor if species variation is increasing in a particular site over time or decreasing over time. This would also become more efficient because of, like I said, the short amount of time, but it also requires less people. On the left hand, we see researchers doing the point count method. There's about 10 people in this picture, and they're all doing one site. This is also very time consuming and can take 6 hours or more. In some studies, it takes multiple days to survey an entire site. On the right is our method. Up to only 5 of us, so 4 or 5 of us went out, and with all the preparation and taking all the photo quadrats, surveying the whole site took around 2 hours or less. And we were also able to find a higher average of species diversity. So this just shows that using photo quadrats can make organizations a lot more effective in surveying the species diversity.
Now with that said, what could we change about this survey to benefit future studies? During our analysis of the photo quadrats, there were some photos that were out of focus, making it very difficult to identify species and the numbers on each cone. In the future, it is important to make sure that photos are very clear so that species can be easily identified and photos can be accurately georeferenced. After the photo has been taken in the field, it is important to assess the quality of the photos and whether or not we can zoom in and clearly see species and the numbers on each cone. While identifying species in our analysis, it was sometimes difficult for us to decipher between species. It was easy for us to determine species in the field based on their physical characteristics, such as color and texture, but these qualities were sometimes lost in our photo analysis. Being unable to accurately identify some species in our analysis may have slightly skewed our data because we were unable to differentiate between similar-looking species, such as hermit crabs or snails. In future studies, we can fix the issue of unidentifiable photos by incorporating advanced technologies such as high-quality drones. Drones can reduce the chance of human error and increase efficiency by taking high-resolution photos of survey sites. Drones can cover a larger amount of area in a short amount of time than manually taking photos of each site, while also reaching areas that are not always accessible by foot. They're also beneficial when trying to gather data during a rising tide or a fast-changing environment. Another technology that organizations could incorporate into their studies is GIS, which could assist in organizing and analyzing data very efficiently. GIS was very useful during our study when we auto-classified imagery and georeferenced our photo quadrats. While drones and geospatial technology have their benefits, they also have limitations such as their high cost and high amount of time spent on training. But as technology continues to improve and become more accessible, we have the ability to open doors to endless research possibilities of all scales. We'd like to thank Dr. Corey Garza and Taylor Eddy for their contributions and guidance during our research, and because without them, none of this would have been possible. And we'd also like to thank the Marine Landscape Ecology Lab for allowing us to use their tools and technology during our surveys. We'd of course like to thank Marine for allowing us to survey these sites, and Garapata State Beach and Hopkins Marine Station for allowing us to use their sites for our surveys, and of course California State University Monterey Bay for their contributions to facilitating our research and allowing us to take this class. All right, thank you. Any questions?
there a way to account for any error, like since it's not on a flat surface and since it's like in a rocky kind of habitat?
That is definitely something to consider. By error,